You're listening to Idea Collider, a show that explores the world of asymmetric learning in biopharma. On this show, I will sit down with pharmaceutical experts and business leaders to discuss how to embrace uncertainty and the different learning styles that follow. I'm your host, Rick Ritacco. Let's get into the show.
All right. Today's guest is Jason Tardio, President and COO of Immunic, a late-stage biotechnology company focused on developing oral therapies for neurologic diseases and with a major emphasis on multiple sclerosis. Jason joined Immunic at a pivotal moment in the company's evolution, bringing deep commercial and launch experience from the MS world, including leadership roles at Novartis and Biogen, where he worked across therapies like Myzent, Plegridy, Avonex. And I think what makes Immunic so interesting right now is that the company isn't just trying to compete in the existing oral MS market, it's making a broader argument that the future of MS treatment may require us to think beyond relapse control and more directly about progression, about neurodegeneration, smoldering inflammation, and potentially even viral biology. And that's what I really wanted to explore today, is that bigger question of what happens if the next era of MS treatment is less about suppressing visible disease activity and more about preserving neurological function over time. And with that, Jason, welcome to the Idea Collider.
Rick, thank you for having me. Pleasure to be here.
And so, Jason, you've been part of the MS treatment landscape for years. And over that time period, MS treatment has largely focused on controlling relapses and inflammatory activity. Do you think the industry is beginning to realize and start thinking differently about progression itself? And that might require a fundamentally different approach.
Without a doubt, Rick. It's amazing to think how far we've come in multiple sclerosis over these last 30 years. 30 years ago, there was no available disease-modifying therapies. It was a disease of what we used to say, diagnose and adios. But there's been huge amounts of innovation, huge amounts of advancements, not only around the disease itself, but the understanding of the biological processes associated with the disease. And of course, we've taken those learnings and applied them to developing wonderful medicines that have changed the natural history in the course of this disease. But we're not done. There are roughly 20 or so available therapies today to treat largely relapsing forms of multiple sclerosis. There's only one therapy currently approved for the progressive forms of the disease. But even with 20 or so available therapies, patients still progress. They still have disability, physical disability, cognitive disability accumulation. And this is for a good reason, because MS is a disease of two biological processes. It's a disease of neuroinflammation, but it's also a disease of neurodegeneration. And so the 20 or so available therapies today, they all largely address just neuroinflammation associated with the disease. And there are two biological processes driving disability accumulation. On the inflammatory side, there's what's called relapse-associated worsening. Patients have relapses, they have flare-ups. Sometimes they fully recover from those relapses or flare-ups, and most of the times they don't. And so you get this accumulation of disability over time. That accounts for about 30 or 40% of all disability accumulation, especially early in the disease during the relapsing forms. But the other 60 or 70% is not caused by inflammation. It's caused by this ongoing neurodegenerative process. And this biological process is called PIRA. This is progression that's independent of relapse activity or independent of inflammatory activity. And the reason patients still progress with 20 or so available therapies today is because, again, all the medicines today are working just on inflammation. And so therefore, they're not addressing the totality of disability accumulation. They're not addressing this PIRA, this neurodegenerative component. So there's a need for new novel mechanistic approaches that address not only inflammation, because that is important, but are also addressing neurodegeneration.
Thank you for laying out the context of the market and where we started 30 some odd years ago and where we are today. When you think about it, why has PIRA progression been so difficult to impact? If we think specifically about that place, obviously we have, like you mentioned, a ton of different approaches to treatment of the anti-inflammatory, but PIRA remains challenging.
Yeah, I think So I think just progression itself, there's a lot we just don't know about underlying neurodegeneration and progression. I don't think it's a single process. The belief here is that it likely reflects a mix of compartmentalized CNS inflammation, chronic active lesions, neuronal axonal injury, mitochondrial dysfunction, a loss of repair capacity. There's a lot of things that go into just progression in general. And when we think about progression and the difficulty around just attacking it or addressing it, again, the more that we understand, the better we are at designing medicines specifically target areas of disease. And so I think there's a lot that we still have to understand. But again, I think the biology around progression is more difficult, and there's a lot we still have to just fundamentally understand.
And just building on that a little bit, when you think about what we do understand or what we believe we understand today about progression, what do you think the current market understanding, misunderstands the most?
Well, I think the market clearly understands the fact and the role that neurodegeneration plays. I think there's also difficulty here is when we just think about the ability for us to appropriately attack neurodegeneration. In order to do this, you have to get into the brain. So we need to ensure that we have medicines that cross the blood-brain barrier, that get within the CNS compartment itself. We need medicines that do more than just suppress peripheral inflammation. They have to influence, again, all of the underlying biological processes associated with neuroprotection, that are associated with chronic inflammation, that's associated with tissue repair and resilience. There are some medicines, like a medicine that we're working on, vidofludimus calcium, that we believe could, for the first time in a long time, be big steps forward in addressing progression, addressing neurodegeneration, providing opportunities for direct neuroprotection. But again, I think that as the disease has progressed, as our understanding around the biology, really first and foremost, we had a thorough understanding around inflammation and the roles that T and B cells played in the inflammatory process here. And I would say that as I think about the disease over time, it was often largely thought about just a two-part disease, a two-stage disease in which patients largely were diagnosed with inflammatory disease that eventually just waned over time. And when that happened, you entered this progressive forms of the disease, hallmarked by this neurodegeneration. But what we know now is that this is not the case. It's not a two-part disease, but rather a disease in which there is neurodegeneration happening from day one of diagnosis. It's largely superimposed by this inflammatory process highlighted by these relapses and other focal MRI activity. But these two processes are overlapped and they're all there from day one. So I think that's a huge understanding that, 30 plus years ago, we didn't quite fully understand. So biology and understanding of the disease is getting better. Our ability to specifically target medicines to address either inflammation or neurodegeneration is getting better, to design those medicines to allow, for example, better CNS penetration and actually get into the CNS to address biological drivers of neurodegeneration has gotten a lot better. And so we remain optimistic that the next wave of medicines, again, like the medicine we're working on, vidofludimus calcium, can perhaps do just that.
Your depth of knowledge, your enthusiasm, and your optimism are always contagious, Jason. So thank you for that. It feels like Immunic's thesis is broader than simply developing just another oral MS therapy. So when we think about where the market's been, where we are today, and all of the different key pieces that you've just laid out, what specific biology do you believe current therapies are still under addressing?
I think it's really around, neurodegeneration, or more so, if it's said other ways, it's about addressing and treating MS beyond relapse suppression. So we know, again, MS progression is not driven only by peripheral inflammatory attacks or these relapses, but it also reflects chronic CNS injury, microglial activation, mitochondrial stress, oxidative injury and stress. And so all of these play a role here. And what's important is finding new novel mechanistic approaches that go beyond just addressing inflammation, addressing relapses and relapse suppression, but also, as I've discussed, really begin to address MS progression and neurodegeneration. And again, that's why we're so excited about what we have here at Immunic Therapeutics.
Absolutely. And do you think that the field is historically over-indexed on controlling visible inflammatory activity because, well, it's simply been easier to measure?
Well, of course, you can measure finite relapses. The patient either has an acute relapse or they don't. You can measure focal MRI activity, like the presence of new gadolinium enhancing lesions or new T2 lesions. These are all pretty objective and straightforward ways to address the disease. As we get into addressing progression and addressing neurodegeneration, that's harder. I mean, the one scale that we've relied on for many years here is a standardized, widely accepted scale called the EDSS scale, which is a longitudinal scale of progression over time in which a patient and their clinician can define kind of at a baseline where that patient sits on the scale. It's from a 0 to a 10, and each of the numbers represent, increased accumulation of disability, largely lower ambulatory physical disability. But this is the scale that we've used. And so when we think about medicine's ability to impact progression and to potentially impact more of the neurodegenerative side of the disease, this is what we've had. But remember, one of the first things I've mentioned is that disability progression and accumulation is driven by both relapses and relapse-associated worsening, but also neurodegeneration. So if you have a medicine that affects relapsing biology, that suppresses relapses, more times than not, you will have some effect on delaying disability accumulation. But you're not going to have a totality of approach here. And so, again, what's missing here is a medicine that does both. And the reason we're so excited about vidofludimus calcium is we believe it will do just that. It is a medicine that offers a dual mechanistic approach. From an anti-inflammatory perspective, it's an inhibitor of an enzyme called DHODH, which is a known, proven mechanistic approach to address inflammatory disease. We take a very selective approach to inhibiting that enzyme, which we think may offer potentially efficacy advantages, but certainly safety and tolerability advantages over a previous first-generation therapy that was non-selective in its approach. But I think what really makes vidofludimus calcium different is that it's an activator of a nuclear receptor, a target called NER1. And NER1, we know, is a target of interest across a variety of different neurodegenerative-related diseases, Parkinson's disease, Alzheimer's disease, et cetera. And so we know that targeting and activating this nuclear receptor may provide clear neuroprotective advantages. And that's why we're so excited about vidofludimus calcium, because we think this dual mechanistic approach will allow us to address not only neuroinflammation, but more importantly, neurodegeneration and progression associated with the disease.
NER1 has been something that I've been interested in seeing effectively targeted for some time. Can you walk us through what does neuroprotection actually mean in practice?
Sure. So NER1, or this nuclear-related receptor protein 1, NER1, it is relevant because it is a target that regulates pathways that are tied to neuronal survival, mitochondrial function, control of neurotoxic inflammatory signaling, right, from innate immune cells. And so this is a very interesting target. As I've mentioned, it's been a target of interest across a variety of different neurodegenerative-related diseases for many years. There's countless publications on the role of NER1 in Parkinson's disease and other neurodegenerative-related diseases. It's only the last probably 10 to 15 years in where it's become a target of interest in multiple sclerosis. First of all, related to the target itself, it's very hard to find a medicine that can bind to and activate Nurr1. And so that's why I think that there's been a slowing of innovation associated with that target over the years. It's not that that there's been a loss of interest. It's just, I think the industry has understood that it's very hard to find a medicine that binds to and targets that target. We know our medicine, vidofludimus calcium, does just that. We have a plethora of preclinical data that shows that it binds to and activates this target in animals. We know that in doing so, we have data in animal models that shows that it prevents neuronal cell death. And so we have a pretty solid belief and understanding here that this medicine is really binding to and activating that target. With respects to MS, what I can tell you is that Nurr1, what we know is that Nurr1 is downregulated in MS patients compared to healthy controls. We know that among MS patients themselves, individuals with lower levels of Nurr1 have more aggressive forms of the disease compared to a matched cohort of MS patients that have higher levels of Nurr1. The most interesting biological link of Nurr1 to MS has to do with pregnancy. Here's what we know. For women who have multiple sclerosis that become pregnant, it's amazing. The body's amazing. Pregnancy is neuroprotective in multiple sclerosis, meaning women who have MS who become pregnant have cessation of disease activity. They don't have relapses. They don't have any new disability progression. They don't have new MRI lesions. It's like during pregnancy, the disease just stops. And a group of researchers out of Italy sought to understand why. And they found that there were 347 transcripts that were differentiated among MS patients compared to healthy controls. And when they followed this cohort of MS patients and a subset of them that became pregnant, they saw that out of these 347 transcripts that were differentiated, 8 of them reverted back to normal levels or healthy control levels during pregnancy. And one of those 8 was Nurr1. So pretty interesting biological link here for the potential that Nurr1 here is not only prevents neuronal cell death and is a neuroprotective target, but is downregulated in MS patients. But then when they become pregnant, you see a normalization of Nurr1 levels in these women that become pregnant. It's a target that we're quite excited about. And we've generated data, I think, clinically now with vidofludimus calcium that supports that there might be something different happening with this medicine beyond just anti-inflammatory approach, but conversely, data that may support that there might be a first real direct neuroprotective approach with this medicine.
Yeah, and it's fascinating to see native biology reflect what we're trying to target and demonstrate with a therapeutic approach. But of course, that has always been the challenge, right? Rats lie and monkeys exaggerate. That classic challenge of having an ambitious biological thesis and taking it into the clinic, which obviously you have had some recent data, both from your phase 2 emphasis and your caliper studies. What have you learned from that? And I guess once you reflect on that for a moment, what surprised you most?
Well, let's take the emphasis trial. This was our phase 2 study in relapsing remitting MS, and this served as the impetus for the design, both dose and ultimately design of our confirmatory phase 3 program, the ENSURE trials, ENSURE 1 and ENSURE 2, that's ongoing in relapsing MS, and we expect data from that in December of this year. But what we learned in the emphasis trial was the following. We learned that in roughly 268 patients with relapsing multiple sclerosis, we learned that both the 30 and 45 milligram dose of vidofludimus calcium provided very rapid and robust reduction in focal or inflammatory MRI activity. And so we had roughly between almost 75% reduction in what's called cumulative combined unique active lesions. We had nearly an 80% suppression in GATA-enhanced lesions at 6 months. And both of those doses, the 30 and 45 milligram dose, were highly statistically significant. So this gives us great confidence that the medicine is a potent anti-inflammatory medicine in providing anti-inflammatory benefits. In that respective study, of course, we also looked at clinical outcomes. We saw, even as early as 6 months, a clear separation between patients on active vidofludimus calcium compared to placebo at reducing confirmed disability progression, with 1.6% of patients on vidofludimus calcium reporting confirmed disability progression at 6 months compared to 3.7% of patients on placebo. So a very clear signal, a signal that you wouldn't expect to see that much of a clear separation that early in treatment. So this gives us confidence of perhaps this uniqueness, this neuron activation really playing a role here on providing neuroprotection, and in doing so, delaying disability accumulation. The medicine is safe, very safe and well tolerated. In that study, we saw a comparable treatment emergent adverse events between the 30 and 45 milligram dose, very low discontinuations, less than 3% of patients discontinuing the medicine. And so we believe we have a clear winner on our hands on the relapsing side. And of course, the phase 3 confirmatory program, the ENSURE trials, will give us additional data. We expect that December of this year. On the progressive side, the CALIBR trial, we released those data about a year ago. This was an exploratory study in a little over close to 475 patients with progressive MS. It was an all-comers study. So we took patients with primary progressive MS, we took patients with non-active secondary progressive MS, and we took patients that had active secondary progressive MS. And the goal here was to give us, as the sponsor manufacturer, confidence that we would see a clinically meaningful signal on the outcomes of delaying disability progression in this tough-to-treat population, and then use those data to help inform what a phase 3 trial could look like. And the results were, we thought, overwhelmingly positive. We saw in the total cohort a 24% reduction in time to 24-week confirmed disability progression compared to placebo. In the PPMS subset, a 31% reduction. In the non-active SPMS subset, a 19% reduction. And in the active SPMS subset, about a 24% reduction. But we also, I think the most interesting signal that we saw was the impact of the medicine in patients with progressive disease that did not have signs of inflammatory disease activity at baseline. And so in our study, for example, in the PPMS subsegment, we had about 16 or 17% of patients that had gadolinium-enhanced lesions at baseline. And so this is a marker of inflammatory disease activity. And so again, in the total cohort of PPMS, we showed a 31% impact at delaying confirmed disability progression. But when we remove those patients that had inflammatory disease at baseline, those 16, 17% of patients, our signal actually goes up ever so slightly. So in the PPMS subset, for example, it goes from 31% to 34% benefit on delaying confirmed disability. In the total cohort, right, of patients, if we remove patients that had the presence of inflammatory disease or gadolinium-enhanced lesions at baseline, that benefit, as you recall, was 24% in the total cohort. So why is this important, Rick? Well, it's important because it signals that the benefits with vidofludimus calcium are extending beyond just inflammation or its anti-inflammatory properties. Because if it was just working on inflammation and just driving benefits via anti-inflammatory benefits, then when we remove those patients from the sensitivity analysis that had inflammation at baseline, then our numbers would go down. And what we see is it actually goes in the opposite direction. So that, to us, signals, we believe for the first time with any disease-modifying therapy in multiple sclerosis, that a medicine is having an impact, a clinically meaningful impact in progressive patients that don't have inflammation, which means and signals that it may be the first real neuroprotective approach to addressing the disease. And again, that's why we're so excited about what we have here with vidofludimus calcium.
Yeah. First, clinically meaningful treatment to stop and prevent the progression. And it really has that neuroprotective benefit. I suppose what's fascinating is that, we have a strong biological thesis on one end, and then we have a very substantial and directional clinical outcome, real world outcome for patients on the other end. When you think about the bridge from one to the next, what are the key pieces of that you would like to see fall into place to make that story fully realized?
Well, I mean, the continued delivery of clinical data is going to be the ultimate signal as to whether or not our hypotheses here around vidofludimus addressing both inflammation and neurodegeneration. So the phase 3 and larger data sets will be confirmatory. And again, as I've mentioned, we expect to have the data in relapsing disease or inflammatory disease this December. But parallel to that, we do plan on kicking off a phase 3 study and initiating that this year in progressive multiple sclerosis as well, based upon the caliber data I just shared with you. That ultimately would give us the end result and the evidence that we need that supports the fact that this medicine may provide a real direct neuroprotective benefit, because if we can show an impact in progressive disease, progressive biology in a progressive patient population, then we clearly will separate ourselves from the vast majority, because the majority of medicines approved today are only approved for relapsing disease. Many of them were studied in progressive disease and failed miserably. So there is a continued need there. I think beyond the clinical data that I've just mentioned, we do have plans to also, generate human data around the effects of vidofludimus calcium increasing Neur1 levels and the effect of vidofludimus calcium on Neur1 levels in human MS patients. I've mentioned we have a lot of data today that gives us confidence in preclinical and animal models that this medicine binds to and activates that target. And again, in doing so, provides and prevents neuronal cell death. I think part of the bridge there is saying, okay, you have this clinical data now that shows that you may have some differentiation on progressive patients without inflammation. So the missing piece is, do we have underlying biology data that supports that the medicine is activating and increasing Neur1 levels, again, in human MS patients? And so we'll generate those data as well. So I think between the preclinical data that gives us strong confidence, are hopefully over the course of the next, let's call it 12 to 24 months, generating additional human data on the role of vidofludimus calcium and its ability to increase Neur1 levels biologically in MS patients, and then paying that off with the clinical data, not only that we've generated to date, but that we're going to continue to generate, I think fills the totality of the story here on not only linking preclinical signals, but also bridging that with some in vivo data in human patients, and then, of course, parlaying that ultimately into a meaningful clinical benefit. So we'll continue to generate those data over time, and we look forward to sharing that as we get the results.
You did raise one of the interesting commercial challenges, though, even with very compelling science, with the strong biological thesis, with a lot of the outcome data, the relapsing MS market is already crowded. It's already very competitive. There's 20 some odd therapies that have approval and are branded. What would make a physician think that vidofludimus calcium is different from existing therapies? And what are you looking to do to drive changes in prescribing behavior?
Well, Rick, we believe it's a misnomer here that the relapsing market is solved for. Of course, it is crowded, but I've mentioned earlier that patients still progress. Our data and the latest data in the public domain suggests that over 30% of patients, even on the most potent anti-inflammatory medicines, which is a class of medicines called anti-CD20 therapies, still have disability progression. And these are medicines that eradicate relapses. These are medicines that eradicate inflammatory MRI activity, yet patients are still progressing. And so if patients are still progressing, it's because, again, this subsequent biological process called PIRA, right? Progression independent of that relapse activity is taking over. So we need medicines that address PIRA, that ideally address both, again, inflammation or RAW, and address neurodegeneration or PIRA. So that's the first unmet need that is clear in the market. So again, we believe vidofludimus calcium's dual mechanistic approach may do just that. But beyond just continued disability progression, there is an enormous unmet need for the development of just safer and more tolerable products as well. I saw a statistic recently that suggests that the number one reason why patients who get diagnosed with this disease the reason that they don't go on a therapy is because of safety and tolerability concerns. Again, many of the medicines today are immunosuppressant in their approach, right? So they
Mhm.
suppress the body's immune system. Given that MS is an autoimmune disease, you're going to have an impact positively on the disease in doing so. But you also, on the other side of the coin, you open up this proverbial can of worms from a safety perspective. So you have patients that have long-term safety issues associated with many of the available therapies, such as serious infections like progressive multifocal leukoencephalopathy and brain infection that can lead to death. You have other opportunistic infections that can lead to hospitalization. You have increased risks of malignancy with many of the available therapies. You have hepatotoxicity, you have lymphopenia, neutropenia, all of these things. And so there's this constant balance between the benefits of medicine may give you balanced with the safety concerns, tolerability concerns, right? A medicine is no good if the patient can't take the medicine, and right? And so there is a need for just safer, more tolerable medicines. And I've mentioned it earlier. We believe one of the potential advantages of vidofludimus calcium is that its safety and tolerability profile, we believe, will be highly differentiated versus what's available today. Much cleaner in terms of just day-to-day adverse events than what's available, much safer holistically than many of the immunosuppressant therapies that are available. And so when we think about this balance act of wanting to drive great efficacy, but balance with low degrees of safety and tolerability risk, we think that will offer perhaps a best in oral class benefit risk profile versus therapies. And then we also have to be very realistic about what's happening in the MS market today, Rick. I mentioned there's two things of note here. I mentioned CD20 therapies. This is a very potent and highly successful class of medicines at addressing inflammation. We talked a little bit about their deficiencies at addressing neurodegeneration. But the reason I bring up this class of medicines is because these medicines, by their mechanistic design, broadly eradicate and deplete B cells. And in doing so, they, of course, have a pronounced impact on disease, but open up people's risk for infections here. And so because there's a lot of patients on this class of medicines, there is a need to begin to think about how do we sequence these patients away from that class of medicines, either once they have an infection or ideally prior to them actually getting a serious infection. How do we think about sequencing them away? And when you sequence these patients off of that medicine, it takes 2 years to replete B cells. So you don't want to necessarily put them on a potent immunosuppressant medicine. We believe vidofludimus calcium, because it's an immunomodulator, not an immunosuppressant, is more ideal as a medicine of choice to sequence those patients on a CD22. I think thing that's happening in the market that we need to really acknowledge is that patients with relapsing forms of MS are staying in the relapsing forms of the disease longer. And this is directly attributed to the fact that we have these medicines that, you remember the first comment I made really out of the gate, that 30 years ago, there was no medicines, but these last 30 years, we've come up with a number of options for patients and treating clinicians. And so what that's done is it's prolonged people to delay what was a previous natural history 10, 20 years after the diagnosis, that you would just immediately get into the progressive, the true progressive forms of the disease. Patients today are staying in the relapsing forms longer. When you have a blossoming segment in relapsing disease that are now 55 years and older, and by our estimates, it's approximately 60% of individuals in the United States with relapsing MS are aged 55 or older. There are just natural biological concerns that you have to think about. There's a natural phenomenon called immunosenescence, in which your body's immune system just begins to wear down. It begins to slow down. This is a natural part of aging. And so because of this, it increases as we get older, it increases all of our risks to infections. And so what you don't want to do is necessarily put this older patient population on potent immunosuppressant therapies, because you're only going to, again, exacerbate this natural phenomenon of this waning of the immune system. And so again, we believe vidofludimus calcium, because it's an immunomodulator, because it's very safe and well tolerated, may be an ideal medicine for these older individuals living with MS, in which, of course, immunosuppressant and immunosenescence is a concern. So in summary here, The relapsing market is crowded, Rick, but those 20 or so available therapies have not solved for the disease. We need medicines that address PIRA and address inflammation. No medicines do that today. We believe vidofludimus calcium may do that. We need medicines that are safer and more tolerated. And we believe the profile to date with vidofludimus calcium may allow us to offer a medicine that is very well tolerated, that may have safety advantages over what's available, and may offer a better benefit risk than any oral product. And of course, as the market evolves here, lots of patients on CD20s, lots of patients older with the disease, there are treatment considerations largely around the idea that you don't want to further suppress the immune system in these populations. And so an immunomodulator like vidofludimus may be an appropriate therapy. So that's why, again, we're excited about what we have here.
I'm very fascinated by the nuance and the differentiation of the modulation versus the suppression aspect, because I think a lot of times everybody wants to be safer, everybody wants to be more tolerable in every market, but actually having that fundamental biology underpinning that story and then delivering against it is tremendous. Otherwise, as you've pointed out, with a long-term chronic treatment for a ever widening segment of the market, as the treatment allows them to age and to, have more health span. I mean, you're really robbing Peter to pay Paul by tackling MS, the inflammation side, but leaving them wide open to the predations of immunosuppression, right? I guess looking forward, since we did start to zoom out there a bit and think about treatment and how it's evolved, and I'd love to take a look at the future state, right? And think a little bit about 5 and 10 years out. What do you think the field will realize it misunderstood about MS today?
Well, I hope that 5 to 10 years out, the market will look very different than what it is today. That beyond addressing inflammation, which the vast majority of the 20 or so available therapies today that, they all do this, and I've mentioned even the anti-CD20s, they do it quite well, that we will have perhaps the next generation of therapies that are addressing more neurodegeneration. And so there's a class of medicine called the BTK inhibitors that have been developed. They've had a mixed bag in terms of some of their data. Some of the trials have worked, some of the trials have not worked. There are certainly some concerns with that class of medicines around the drug-induced liver injury and liver toxicity, some of the infections and infection-related deaths associated with that class of medicines. But there is some data to support. We have to, give credit where credit is due. There is certainly some clinical data to support that those medicines are working beyond just inflammation, that they are getting into the central nervous system and perhaps addressing more neurodegenerative aspects of the disease. Of course, as we've discussed around benefit risk, there will always be this playoff on whether or not that medicine is appropriate for patients, just given some of the safety signals. There's already been one medicine to date called tolebrutinib from Sanofi Genzyme that was denied by the FDA. They received a complete response letter late last year, and that complete response letter was predicated on the risks outweighing the potential benefits of that medicine. So we'll have to see what happens with the other medicines in the class. But I bring up that class because there at least is some underlying data and biologic rationale to support that those medicines may be going beyond just inflammation, which would be obviously very helpful for individuals living with this disease. We've talked about vidofludimus calcium here and its effect on Nurr1 and activating that target and what that may mean for neuroprotection and its impact on more progressive and underlying neurodegenerative biology. So if we can continue to show success in progressive patients, my hope is that in 5 to 10 years, you're going to see additional therapeutic options that are addressing, again, underlying neurodegeneration and biology. I think the ultimate goal here, if we go out to 10 years, but at this point, probably beyond 10 years, is to find medicines that actually just ultimately halt and stop disease progression. No medicine today does that. We do a pretty good job of altering the natural history. We reduce relapses, we reduce new MRI lesions, we delay accumulation of disability, but we don't stop the disease. And so that will be a continued target here as we think about innovative and continue to advance therapeutic intervention here. And then, of course, not only stopping and halting the disease, but perhaps reversing neurological damage that's already happened. This idea around remyelination, right? MS, we know, is a disease of demyelination. These T and B cells, these immune cells, they get into the central nervous system. They attack the myelin sheath, this fatty substance that protects the neurons, right? And so there's demyelination that happens with this disease. And so can we actually augment the body's natural ability to remyelinate through pharmacological intervention? I think I'd love to see that in the next 10, 15, 20 plus years as well. So we still have a ways to go. But the beautiful thing here, Rick, is that there's been a lot of progress these last 30 years. So individuals getting diagnosed today have a much different prognosis if they get on an appropriate disease-modifying therapy than they did 30 years ago. But the unfortunate thing is that we have not solved for it. There's these unmet needs that we've talked about. This needs to address the totality of progression, the needs to provide medicines that are safer and more tolerable, the needs to address specific subpopulations, older patients, et cetera. So there are still needs here. And again, that's why we do the work we do every day at Immunic Therapeutics, because we believe our medicine will help to address many of those unmet needs, but our work is not done. And we have lots to do as we move forward into the future.
I love the way you laid that all out. I think it's interesting to think of it visually as being able to, over the last 10, 15 years, slow the accumulation through our approaches to anti-inflammatory and stopping relapse. Now we're getting to the point where we're flattening the curve. And then I think as the way you just laid out, when we think about remyelination, could we actually return that curve back down to a normalized state? Which begs the question, as you look out and from your perspective, in 10 years or so, 15 years maybe, are we still thinking about the terms of relapse versus progressive, or are we characterizing the disease in an entirely different way given the new therapeutic approaches available?
And I think that there's experts, and we were with a series of advisors recently, many of which Dr. Fred Lublin out of New York, who has really been the pioneer of the classification system associated with MS. We are having these discussions. I mean, look, I think that it's well understood today that MS is not a disease of relapses and inflammation that eventually manifests into a disease of neurodegeneration and progression. I think it's well understood today that this is a disease of both neuroinflammation and relapses and a disease of neurodegeneration and progression from day one. And the questions here around how we classify the disease, how we classify subsets of the disease, we're trying to advance our understanding of just the biology itself. We have a ways to go there, as we've discussed early in our conversation today. And some of this is also regulatory driven as well. This is how the regulators want us to look at these diseases and the subsets of the disease. We'll continue alongside, the different advocacy organizations like the National Multiple Sclerosis Society and others. We'll continue to advance our thinking, but also pressure test the regulators and how they think about the disease. Look, there is some good biological rationale to think about, for example, progressive disease as one disease beyond just a disease that's a disease of patients that have labeled primary progressive multiple sclerosis, which is a diagnosis in which patients never formally had inflammatory disease. They got diagnosed just with a disease of this constant slippery slope of physical disability accumulation without these hallmarked relapses, versus secondary progressive MS. This disease is a disease in which individuals were previously diagnosed with relapsing forms or inflammatory forms of MS, which just over time, they've morphed now into more progressive forms of the disease. And so if you have a patient that has secondary progressive MS or non-active, non-inflammatory secondary progressive MS, the biology is probably not that vastly different than a patient that has primary progressive MS. And so then the question remains is, how could we and should we study these patients? Can we do a basket study in which you're looking at both SPMS and PPMS patients? Because if the biology is relatively the same, can we just lump these patients into, to progressive forms of the disease as opposed to these subsets? But we're continuing to advance our thinking there. And again, I think that suspect perhaps at some point, we will get to the point where we have aligned, at least biologically, that progressive disease is progressive disease. Whether a patient got there via secondary progressive MS because they one day had relapsing disease that is now, the inflammatory process has waned, or whether they got there because they were originally diagnosed just with primary progressive disease, it's probably moot. They are now in this progressive stage of the disease, and the biology is probably, if not identical, is probably generally similar. Let's treat them as such. And then perhaps we've now advanced our thinking to say, all right, there's a medicine hopefully to address underlying progressive biology, regardless of whether or not the patient has PPMS or non-active SPMS, it's progressive disease. Mm-hmm. So that's where I hope and believe we'll get to, hopefully sooner rather than later, Rick.
I'll borrow some of your optimism, and because I think that would open a lot of avenues for more targeted and more specific approaches for the patients writ large, rather than trying to narrowly focus them in some of these, the relapse and progressive buckets that we have today. Let me again, with one final question, I'll borrow your unbridled optimism. When, not if, when Immunic succeeds, what changes for patients that current therapies still struggle to protect?
The important thing to remember is that if vidofludimus calcium is successful and our hypothesis about this medicine is spot on, and the data that we've generated to date continues to hold true, we think this medicine could really change the treatment in multiple sclerosis, right? First of all, relapsing disease, because you'll be addressing both underlying biological processes that drive disability accumulation. And so we may offer the first unique and differentiated approach to address the totality of disability accumulation. But even from a simplistic perspective, this medicine seems to have advantages from a safety and tolerability perspective. And so it is important that we have patients on these medicines. And if the biggest driver for patients not starting or not being on a therapy is safety and tolerability concerns, then the fact that our medicine seems to be safer and perhaps offer advantages from a tolerability perspective, that alone could really change relapsing disease, because all those patients that are not on therapy today or may choose not to go on therapy after an initial diagnosis, perhaps they'll reconsider given the profile of our medicine. And then, of course, as we advance our studies in progressive disease, there's just enormous unmet need there. And if we can deliver on the data that we've delivered to date in a confirmatory progressive population, I believe vidofludimus calcium will be the cornerstone of care in progressive disease, a medicine that is truly offering direct neuroprotective benefits via this neuron activation, a medicine that is an immunomodulator, not an immunosuppressant. And remember, progressive patients are usually a little bit older with comorbidities, more advanced disease. And so you are concerned about immunosuppression in those patients. So it could offer distinct advantages there, and it's safe and well tolerated. So why wouldn't it then be the real cornerstone of care? I believe if we deliver on our promise via a confirmatory phase 3 program in progressive disease, I have no doubts, if we do part 2 of this conversation, Rick, 5 years from now, we'll be in a situation in which we're talking about vidofludimus calcium really being the cornerstone of care in progressive disease, a medicine that potentially has changed the face of progressive biology and progressive disease in general. So that's my hope here. Let's keep our fingers crossed.
Jason, I'll look forward to that next time we sit down in 5 years. And thank you again for your time today, for you and the hard work of the Immunic team on changing the face of MS treatment. And look forward again to sitting down with you in a few years and going through how the world has changed. Appreciate your time today.
Yeah, thank you again for having me.
Of course. That was Jason Tardio, COO and President of Immunic.